Okay, Sam, thank you once again.
Thanks, man.
Now, Anne Van Leer from the US Naval Academy is going to introduce Stephen Schiff.
Good morning. It's my honor to introduce Dr. Steve Schiff. Dr. Schiff is the Brush Chair Professor of Engineering and Director of the Penn State Center for Neural Engineering. He is a faculty member in the departments of neurosurgery, engineering, science, and mechanics, and the Department of Physics at Penn State. Dr. Schiff is a pediatric neurosurgeon with particular interests in epilepsy, hydrocephalus, sustainable health engineering, and global health. He holds a Bachelor of Science degree from MIT and a PhD in physiology and MD from Duke University School of Medicine. He wrote the first book on neural control engineering, published by the MIT Press in 2012 and soon to be in its second edition. He serves on too many editorial boards. He's a fellow of too many organizations, and in his limited spare time, he plays the viola in the Nittany Valley Symphony in an, I quote, out-of-tune manner. However, even given his limited time to practice, I suspect that that will soon be further reduced, as he has recently been named the recipient of the NIH Director's Pioneer Award for Research. Please help me in welcoming Dr. Stephen Schiff.
Thanks very much, Anne. Wish Mom was around to have heard that. It's always the question, will it work? Great. Well, thank you so much to the organizers of the meeting to invite me to talk. And let me tell you a little bit of a story of some work that we think is quite interesting. And also, please remind me never to try to follow Sam Berkowitz in any kind of lecture. I'd much rather hear Sam than myself right now. Seizures were first recorded, to my knowledge, in 1933 by Hans Berger. This is quite the tracing, actually, if you're a fan of recording seizures. It's got a beautiful DC component to it, recorded from the scalp. We're very hard-pressed today to do as well. And a few years later, Aristides Leon in Harvard University was studying seizures in the cortex of the cat. He had an array of electrodes put down, and he has trouble finding seizures. Instead of Berger's epileptic events, he's watching this wave of cortical silence progress. And of course, his failure turns out to be a major discovery called spreading depression rather than excitation. When I was a graduate student, spreading depression was, I'll call, a bit of a backwater of neurophysiology. The good people did seizures, and, well, there were these odd people who did spreading depression. But we came to realize in the last several decades that spreading depression is very likely the physiology that underlies auras and migraines, and most recently, especially through rather heroic efforts of people like Jeff Harding, we're realizing that spreading depression is a major factor in how well your brain does after stroke, hemorrhage, traumatic brain injury. In the audience, you folks know very well that many of your anti-epileptic drugs work quite well in certain people with intractable migraines. And of course, as Sam has started to discuss, we do have families where we see patients with both seizures and migraines. The most famous of these are the variants of familial hemiplegic migraine, and again, with some of the genes that Sam just mentioned, such in sodium channel, calcium channel, and ATP. Keep this in mind because we're going to talk here about some modeling that attempts to show why the ATP pumps are so critical. And the best person who's talked about these variants, who I've learned a lot from, is Mike Rogowski. So what else do we know? If you go into the lab, I used to watch Ray Dingledine and Steve Trainalis do these experiments. It was very exciting. They'd take slices of the brain of a volunteer animal and they put it in some perfusion solution that had a little bit extra potassium in it. And these things would generate what looks all the world like epileptic seizures, like clockwork, every minute or so. And then other people were not satisfied with the amount of potassium, so they went exploring putting in about 2, 3, or 4 times as much potassium. This is David Andrews' lab and Brian McVicker's lab, both in Canada. The Canadians love to add a lot more potassium. And they discovered that you could get periodic spreading depression rather than seizures from the same thin slices of brain in the dish. Hodgkin and Huxley put together our first model of propagation of an action potential by studying the squid giant axon. And those of you familiar with this will smile, and those aren't, don't worry, I'll save you. The Hodgkin-Huxley equations, as one of my faculty members in college once reminded me, was the first and last good work in computational neuroscience. These things demonstrated that they could account for thresholds and accommodations and oscillations and refractory periods, and they left out ion fluxes back and forth in the membrane, energy. They didn't have seizures or spreading depression, but they're an interesting model. I look at this and I ask, well, this is kind of a perpetual motion machine, and that's not good. And they assumed that this was an infinite neuron in an infinite volume, which also doesn't fit. Oh, and it spikes. It spikes very well. So what else can they do? These experiments have been done for decades now. So starting with Speckman's work in Germany, using oxygen-sensitive microelectrodes. These are polarographic electrodes, and you can show when there's a seizure in the brain of an animal, the local oxygen pressure plummets. You can show in the dish, this is Sonia Behar and Ted Schwartz's work, the same phenomenon happens. So, okay, energy might be important. Beautiful work from Megan Nedergaard's lab in the last couple of years have shown these exquisite rings of hypoxia around individual brain capillaries during spreading depression. Spreading depression also chews up a lot of energy. And then if you look in the literature, you see these strange things. Nobody wants to publish what's on this slide because we've all been taught that seizures or spreading depression are different phenomena. Mike Haglund and Phil Schwartzkrein published some things like this. What's that? Kind of a seizure and with a spreading depression followed by a seizure. This is Gaberchek's work with George Samjic. The first half is a seizure and the last half is a spreading depression-like phenomenon. So I call these mixture states. We have in our lab for years looked at, we thought, well, seizures, they must be pretty easy to figure out. You just stick enough electrodes in and torment your graduate students and postdocs, and then the pattern that accounts for seizure patterns will be obvious. If only it worked that way. One of the things we have seen, this is amazing experimental work from Jokubas Zuberkis, is that interneurons, and we saw this the other day in some of the work that Sid Campbell had displayed, can fire preceding the onset of a seizure. They can stop firing what looks like spreading depression. When they stop firing, and these are part of local subnetworks of bound together interneurons, you'll see the pyramidal excitable cells release. They generate a high-intensity runaway excitation of a seizure, and on occasion you'll actually see these networks oscillate back and forth with spreading depression, seizures, spreading depression, all in nearby single cells interacting with each other. And on some lucky days, you'll stick a few interneurons, and they all go into spreading depression together. I don't know quite how to describe this in terms of the old theory that seizures are simply hypersynchronous activity, I'm not even sure what the synchrony is and things like this, unless you want to start describing the synchrony in the spreading depression when the seizures are happening with the neurons are. So what could you add to the Hodgkin-Huxley view of spiking in neurons to try to account for some of this? Well, you could say that in addition to the currents that flow in the membrane, sodium, potassium, and chloride, that's the original Hodgkin-Huxley, you could add the pumps. The first person to do this, I mean, neurons store energy. The atmosphere around Melbourne has been doing a wonderful job of dissipating a lot of its energy almost the last week. So storms and rain showers are a way for energy to be dissipated. If you like to think of seizures as storms, they dissipate this stored energy. The first person to really study how changes in potassium induce oscillations in certain regions of a membrane in the brain was Maxime Bazanoff, working with Terry Sejnowski, and we've also produced a number of studies of these dynamics over time. And then we finally started looking at some better ways of looking at oxygen when brains seize. I had a wonderful student named Justin Ingram who would go into the lab for months and sort of tinker. And he ended up inventing a range of new technologies. I won't get into the details. This is a fret-based dye that uses a quantum dot to transfer electrons. It's a really cool thing if you're a chemist, but he spread it all over cover slips and could look at different light ranges and look at how the center of these pieces of brain are more hypoxic than the edges, of course. And by coating electrodes during seizures, you could show the differences in nearby layers, which is really important in orchestrating the patterns that you get out. And on some days, he'd backfill a very small electrode, and we'd snuggle it juxtacellularly right up against neurons. And you could show the oxygen depth after the seizures had occurred. So how could you add oxygen dynamics? You might say the rate of change of oxygen is dependent on the pumps in neurons and brain that chew it up. And then you could say, well, it diffuses in. We know the basic bulk diffusion properties of brain to oxygen and other ions. And then you could go into the biophysics, and you could find that indeed these pumps have a sigmoidal relationship in how well they pump to how much oxygen is around. You starve them of oxygen, they don't do anything. So you could then go back in and you could adjust oxygen pressure around brain that was not seizing, and you find this band colored here in sort of a red light color, where as you begin to drop oxygen pressure, you see seizures. And if you drop it too much, there's just not enough energy around to support seizures. I guess that's not surprising either. And if you go back to that model with the two pieces, the pump current and the oxygen that has to be supplied from a capillary or a bath, either way, you drop out a range of lowering oxygen pressure when you see seizure activity. It's one of the few things I've ever done where I've never tuned it. We just kind of threw in the two pieces of biology you knew were there, and it comes out ridiculously in the same range that you see the oxygen pressure drop in experiments, which proves it's better to be lucky than good. And there are, at this point, we began to ask if, just as the biology teaches us from our patients with both conditions, is there a way to unify seizures and spreading depression in the standard models we use for brain excitability? And there are three lessons in that. The first lesson is that extracellular space matters. This is an interesting group because there's the epilepsy community here, and then there's a bunch of us from the seizure prediction workshop, which are focused on things like computational models. Most computational models of neurons and brain ignore the kind of nasty little fact that your neurons are surrounded by an extremely small space. Hodgkin knew this. 4 years after they published the papers that they received the Nobel Prize for, they did this experiment, which is not nearly as well read. In all the initial experiments they did before World War II, they stripped the glial sheath that surrounds that squid giant axon off and laid the axon in a chamber. And in these experiments, they left it on. This is some of the very early electron micrographs. And what they showed is that every time they fire a single action potential, you squirt about 1 millimolar increase in concentration of potassium around that membrane. They did other experiments where they would repeatedly stimulate it, and the increase in potassium is enormous. That changes the excitability of the neuron. The second thing is that conservation matters. It turns out in retrospect that, if you study the brain, you never study the heart. And the heart physiologists have looked at this question a bit, but of course we don't speak to each other and we don't read their literature. I'm sorry about the complexity. You can be an accountant in looking at models and actually count the number of sodiums going in and potassiums coming out and keep an accounting of the ions. Computer time is cheap, so why not do that now? And lastly, size matters. Because if you're keeping an accounting of the ions, you can calculate osmotic pressure. Well, you do that. If you went to med school, you throw out all the complexity of osmotic pressure, you just add up all the positive and negative ions, and you get the pressures that you need, and you then let Vince Watkins in the audience, I see. So we stole this formula from his gorgeous work that he's done for well over a decade on such phenomena. So here's the difference. In the original formulation, we took a leaky nerve membrane and we had 3 ions. We're then going to add to that an extracellular space, glia, that buffers these ions, pumps the chew-up energy, a diffusion pathway, first just for potassium and oxygen, but we're working on putting more things in. And the thing no person ever wants to worry about in the brain, Hodgkin and Huxley hated it, we didn't want to do it either, is account for chloride. But chloride is critical in the models. They leak, and they don't behave normally if you don't put it in. In fact, Sam just mentioned genetic issues with the chloride transporters that are important to some of the genetic causes of the epilepsies. Realize, of course, that in those genetic variants of epilepsy that Sam just discussed, a lot of those genes are represented in the model I'm about to discuss with you, and can be so studied. Lastly, we'll let the neuron shrink and swell. This turned out to be much more important than I ever guessed. So what do you get if you actually now raise potassium on this potentially unified and expanded model of the Hodgkin-Huxley equations? You find that there are 2 regimes of oscillations. One is where we find seizures. There's a physiological ceiling. In brain that has seizures, we never see the potassium go much higher than 12 to 15 millimolar in real life. And then there's a broad regime where you see spreading depression. I like the color plot of this, but if I smush it down onto a plot of oxygen and potassium, you'll see that there's a broad region where there's a steady state. We'd like to call a lot of that a normal operating regime in the brain. There is a region of intractable seizures. There is a region bordering it of spreading depression, way down low without enough oxygen. I like the phrase of Michel Le Van Putten, the wave of death occurs. And we can show in the model that happens. And we'll simplify it just a little bit in order to study how it behaves mathematically. And that's important. Look, nobody wants to go and use something this complicated and this messy other than to gain some understanding. And you'd like to say whether these kinds of transitions into seizures and migraines have an easy to understand underpinning It provides the framework for the transitions that we see in patients. And in fact, if you do that analysis, you find that the models have two regions where they oscillate. You can go back into the full model and show the prediction that at very high values of potassium, you'll shut off the spreading depression. Importantly, the model will show unity. As you drop oxygen pressure, all of you are sitting there in the audience, and if I could test around your neurons, they're all getting bombarded, hopefully with a lot of activity. They're not spiking all that much, but they are spiking enough. And the oxygen pressure around the majority then is lower than what you have in your capillaries. You can find unity in the oscillations as you drop the oxygen pressure in both this reduced model and the full model, and probably in all of you sitting here. So what does that tell you about what we know about seizures and spreading depression? Mind you, this is a model like Hodgkin-Huxley of a little patch of membrane, a neuron. And Hodgkin and Huxley's dynamics of resting state, raise the sodium conductance a little, you'll get steady spiking. Go back to rest state and stimulate it, you'll get this Hodgkin-Huxley spike. It lives right here. It lives at normal oxygen and low potassium. If you crawl along and raise potassium, you repeat the Dingle-Diamond and Treynellus experiment. Keep raising potassium, you get out what Andrews and Anderson and out here with Brian McVicker have found at higher levels of potassium. If you drop oxygen right into here, into this region, you demonstrate, remember that chimera of seizure and spreading depression I showed you from Czech and Samjan's work? It seems to live right in here, and they've actually lowered oxygen pressure to get that. Here's the wave of death with Michel Le Van Putten and Zan. You get a lot of physiology out of this kind of framework. If you've noticed, and you're a fan of spreading depression, this little spike. Very few people look at spreading depression in single cells. You're used to seeing this as a smooth curve from your measurements if you're an epilepsy expert. But if you go into the marvelous electrophysiology of Oscar Herrera's, you'll see this spike all the time, because that's what a single cell does when it goes into spreading depression. There's an awful lot that seems to drop out of this. Most recently, we just published this. We looked at volume. So all of you have probably manipulated humans. You adjust their osmotic pressure. We all use mannitol in patients who need to have their brain shrunk a bit. Volume serves in this model to also get you in and out of seizures and spreading depression by itself. And if you let the brain free run, you will see it take a trajectory through different volume regions. The point here to summarize is that the seizures occur at a different cell volume ratio to the extracellular space than spreading depression occurs. And just very briefly, I think we're beginning to see what might help us explain the physiological ceiling that potassium doesn't go above in our patients with seizures, and a very different ceiling that appears to separate spreading depression from anoxic depolarization. And again, these are inferences from our theoretical work, but they're very testable in both humans and our animal experiments. So what does this show you? Unification is what we would call an emergent property. We never went looking for this. I was trying to improve the model for spreading the depression we had so that we can use some control engineering principles called model-based control in real time to control migraines from controlling spreading depression. So this is pretty easy to do, and if any of us knew we could do this, we would have done this decades ago. We just threw in what we knew seemed like biophysical basis. There's no empirical model that we would guess at that would do this. And it brings up some very interesting questions for control. if I have a plot like this of basic physiological variables, which will vary depending on the genetics of the channels that I have, I'd like to stabilize it here. I don't want to go wandering into intractable seizures and spreading depression and trying to do something once it starts. There's a growing and very interesting literature on observing when you're close to a stability boundary and trying to control from that. It was brought up a bit yesterday in the seizure prediction using some of Jeff Gore's fabulous work with yeast dynamics in vitro. But we're working on using this kind of model framework to ask if we can stabilize the system, what people call neuromodulation at times now, rather than having to stop seizure activity. It also brings up the interesting prospect that if you're, let's say, in a spreading depression episode, how you control the system might be very important. You want to bring it to the point where you're back in a relatively normal operating regime. I don't want to take someone with a migraine and push them into intractable seizures, which seems quite doable from what this would. It also lets you talk about brain and controller energetics. We don't want to be changing the batteries in control devices for seizures as frequently as we appear to have to do. The other thing we want to worry about is I can overdrive your neurons and kill them. And you want to be able to model the energy load you put on neurons in any kind of stimulation device. I chatted with Vitsa Wadman at a poster at the Society for Neuroscience about 15 years ago, and I asked him on his new work in modeling spreading depression, would there be a polarization axis that we could try to control? And he said yes. So 15 years later, I can stand in front of him in a talk and say we're actually using some of the things that we've learned about controlling brain and stimulating it electrically. This is a diagram of one of the brilliant electronic devices that Bruce Gluckman, who's also here, has worked on. And we're showing now that we can actually break up some of the rings of spreading depression using very small electrical currents. Maybe by next year, we can actually report those results. So a lot of people have worked with us on the things I've discussed today. The three most important trainees were Vinny Ula, who's now at the University of South Florida in physics. Yina Wei is at University of California, Riverside doing a postdoc. And Justin Ingram is at the Penn College of Technology. He's also on faculty. We put code in an archive. If any of you would like to work with this, it's all available with a link in the most recent paper. And I thank you very much.
Thanks, Steve. That was really spectacular. Questions? Hilda.
Yes. I want to know a little bit about how you completed some of this. The balance of oxygen or potassium that you have taken, have you treated those, the interaction with them in a phenomenological way, or you have considered them as oscillators with a certain frequency distribution and do some quantum mechanics?
No quantum mechanics, but we're pretty simple. You have a tank of oxygen, you bubble it through the solutions, and you can actually, I'm unfortunately doing this for too many years, you can bubble it to a certain concentration. The other easy way to do it is you just kind of mix up in the same solution one that has nitrogen and carbon dioxide rather than oxygen, and you flow them together, and then we measure the oxygen pressure in the chamber. So it's like doing any other physics experiment. We control individual variables. Everything else with the pH and things are buffered. So we manipulate both oxygen and the potassium that the tissue is exposed to in order to do the experiments. And then we try to model those biophysically. You just use ordinary differential equations to do it. These are single compartment neurons. So if all of you are sitting there cringing at that thought, it used to be if I talked about this, people would complain. We can't see seizures in single cells. Fortunately, people like Vitsa and George Samjian and Barry Connor have beaten everybody up to the point that you don't complain anymore. But we've not studied these principles in extended neurons yet. That's critical to do. And of course, seizures occur in networks, and spreading depression occurs in an extended medium with reaction diffusion dynamics. And we haven't yet, we're starting to, but we haven't yet looked at it in those more realistic frameworks. Any?
I think we've got time for one more question from Chris there.
Absolutely, thanks. Fantastic question. Just a question, doing brain slices all these years, we just get some saline, up at saline and carbonated, 95% oxygen. Some years ago it struck me, that's a funny oxygen concentration, isn't it? 95% oxygen dissolved. What's your take on what we're doing in the brain slide versus the real system?
Oh, absolutely. Great question. And of course, what you're getting to is that the solubility of oxygen in any kind of aqueous solution is much less than the oxygen carrying capacity of blood. And we certainly recognize that. I think it's a phenomenon of scale. We've looked at a wide variety of oxygen pressures, but you need to be able to translate that capillary bed, where, again, in the measurements that have been done, we see that there are these cones of oxygen pressure and oxygen concentration going from the typical intercapillary distance. People love to complain about what's the right number to use, but let's say I'll quote between 25 and 50 microns. It means a lot of your neurons are a fair number of microns away from the nearest capillary. They will be running, just like the Nettergaard work just showed you, they will be running at a much lower oxygen pressure than the neurons that are just sucking from the oxygen supply right next to the capillaries. So the problem with real life is we'll see a whole spectrum of this occurring, and we, at present, don't have the technical tools to actually look at that as a function of space and time when the neurons are active. But I think the optical techniques that Nettergaard get us close, and it's all very, I haven't gone into some of the details, but that work is very consistent with what we see in the slices and what we've been modeling. Thank you all.
Thanks again, sir.